our webinar speaker, Dr. David Leibly. Dr. Leibly is a biochemist and structural biologist. His work at ADRX is focused on the discovery of novel inhibitors of protein aggregation. This protein aggregation is most commonly associated with neurodegenerative disease, such as Alzheimer's and ALS. His current work seeks to design and select highly specific peptides to be used as tools to facilitate small molecule discovery. Dr. Leibly earned his PhD in the Todd Yeats Laboratory at UCLA, where he designed and determined the structure of oligomeric forms of GFP as a crystallography tool. He has 15 years of drug discovery experience, including structure-guided protein kinase projects, novel antibody discovery, and peptidic amyloid inhibitors. The title of his presentation is Identifying and Targeting Unconventional Druggable Sites on Pathological Amyloid Fibrils. David, I'm handing the cursor over to you. Please take it away.
Great. Thank you very much for that introduction. And thank you all for attending this webinar today. As Carson said, I'm going to talk about a little bit about the work we do at ADRX. And just for a little bit of background, we are a small preclinical biotech company based in Thousand Oaks, California. And since our founding in 2013, our mission has been to find novel inhibitors targeting these amyloid diseases. And as mentioned, I think the most common of these are neurodegenerative diseases. And one of those, the most common, is going to be Alzheimer's. Alzheimer's is linked to the protein tau. And on this title slide here is actually the atomic structure of a tau paired helical filament that was solved from fibers that were extracted from the brain of a deceased Alzheimer's patient. So we're looking at the end of it here, and really this talk is focused on how we go about designing inhibitors that bind to this displayed surface here to arrest amyloid aggregation in hopes of arresting disease progression and ideally reversing disease progression. I've split my talk into 3 parts today. In the first part, I'm discuss amyloid disease and the structure of these amyloid proteins as a whole. Moving to part 2, I'm going to discuss how we design and evaluate our amyloid targeting peptides and how we utilize peptide microarrays for that now. And then in the third part, I'm going to discuss how we leverage those targeting peptides for the discovery of small molecules. Amyloid-like diseases is a large and growing field currently. There are several diseases that present in very different ways, have very different characteristics. However, many of them, all these amyloid diseases are characterized by very, very specific protein-protein interactions. These protein-protein interactions cause aggregation. It can also cause condensation of proteins, and this leads to disease. So many of the diseases were previously considered unrelated, untreatable, but they all have a fundamental structural basis that we're targeting at ADRX. And here in this cartoon, we're illustrating some of these diseases. And these are human amyloid diseases. So, so first is going to be up at the top here. This is going to be neurodegenerative diseases. Again, this is going to be your Alzheimer's, ALS, Parkinson's. Here, these are all in the brain, typically intraneuronal, and the mechanism of disease is kind of poorly understood. But what is known as protein in the neurons is capable of, in some instances, condensing, and these condensates can turn into amyloid, or nucleating amyloid itself. These amyloid these amyloid fibrils that our thought and our hypothesis is that they cause dysfunction to the neurons and cause disease. Now, these are primarily intraneuronal, but also in some instances, some of these proteins can be extracellular. And I think the most common extracellular protein that has been linked to disease has been A beta with Alzheimer's, but also upon neuronal death, if it had interneuronal aggregates of protein tau, those will then become extracellular, they can propagate to other cells. And these are really the hardest to target of amyloid diseases, and are characterized by, and I've broken them down here by a few different categories. Tauopathy, so diseases that are linked to the protein tau, that's Alzheimer's, Pick's disease, frontotemporal dementia, ALS, which is linked to the protein TB43, synucleinopathies, linked to the protein alpha-synuclein, which Parkinson's and Lewy body dementia. These are all neurodegeneration, all occur in the brain. Next is going to be systemic amyloidoses. Now, these are a little different. These amyloid diseases occur from the aggregation of typically well-folded proteins, such as the transporter protein transthyretin, or TTR, that undergoes aggregation and causes transthyretin amyloidosis, or IgG light chain, which causes light chain amyloidosis. Here, it's a more clear link between fibril formation, amyloid formation, and disease, where aggregated protein actually deposits into organ tissue or peripheral neurons and causing dysfunction to those. Now, the last human biology of amyloid that's known today is going to be cancer. So this occurs typically by misregulation of the cell cycle by proteins either forming bimolecular condensates or aggregates that disrupt the cell cycle or lock tumor suppressor proteins such as p53 into aggregates where they can no longer function. And these all on the surface seem to be very, very disparate diseases, but all have a fundamental basis in protein-protein interaction that we target in our work. Now, to briefly talk about neurodegeneration. So the mechanism of disease, in many cases for neurodegeneration, is kind of poorly understood. And there are many hypotheses for those, for the cause of disease, whether it is the aggregates themselves causing dysfunction, whether there's a toxic oligomer or prefibrillar species that causes toxicity leading to neuronal death, cell death, or whether just the presence of these aggregates actually spurs some sort of immune response that causes disease. But fundamental to all of those is the presence of aggregated protein. And we hypothesize that by preventing the further aggregation of protein, or by stopping the nucleation of aggregation, we can arrest disease progression. And this illustration of neurons is what we believe is happening. We have a healthy neuron on the left. Through some event, aggregation starts. Aggregation somewhat progresses unchecked because of continual expression of proteins such as tau, to eventually cause neuronal death and neurodegeneration through cell death. And then upon cell death, you have propagation of the amyloid protein aggregation to neighboring cells. So this is what we want to stop. We go about this through structural-based approaches, which has really been enabled through some groundbreaking work from one of our co-founders, Dr. David Eisenberg from UCLA. In the late 1990s, it was really unknown if any of these amyloid had structure. Many of the proteins, especially for neurodegeneration, such as tau, alpha-synuclein, they are inherently disordered proteins. And it was unknown if upon aggregation, if they're forming just amorphous aggregates, which would be very, very difficult to target, or if they're forming ordered structures that can be targeted. In the early 2000s, Dr. Eisenberg really set out to see if there was a structural basis in disease. And he found that there was. In 2005, the first structure, dubbed a steric zipper, was determined in David's lab. And here, the adhesive segments from one protein was synthesized as a peptide and crystallized and solved through a new technology at the time, microcrystal X-ray diffraction. And that really revealed that there is a structural basis, a well-ordered core to these proteins. This work spurred in 2010 the creation of its database known as ZipperDB. And through this database, the prediction of these adhesive segments was possible. We found that many proteins, especially these proteins related to neurodegeneration, have the potential to form these structures via adhesive segments. Around the same time, microcrystal electron diffraction, so using electrons in an electron microscope to solve the structure through diffraction, was developed. We had a kind of a revolution of even more structures being determined. Fast forward to present day, recently with cryo-EM advances, we now have the structures of fibrils, such as the one on my title slide there of tau, of the pathological proteins extracted from the brains of individuals, which has really opened up the world, or at least for us, of how we can target these amyloid proteins with inhibitors. Now, as I said, these proteins are highly ordered, and here are a few of the structures here. So there are two flavors of aggregates I'm going to kind of talk about today. The primary one is going to be amyloid fibers, and that's depicted on the left here. So on the far left, this is really an illustration of what one of these fibers looks like, one of these pathological fibers of tau, known as a paired helical filament. So what's formed is two monomers of tau that adopt this J shape, form a dimer, and these dimers of tau stack and grow as the fibril. So here's looking at the cross section of that. So the two monomers form this J shape with a dimeric interface in the center. The second form of aggregates that I'm going to briefly discuss is going to be biomolecular condensates. Now these are very, very similar to amyloid. The difference is the backbones of the peptide backbone is actually kinked, where in amyloid we see straight backbones that form very, very stable and irreversible interfaces, colored in red here. The biomolecular condensates have a kinked backbone, and this kinked backbone creates a less stable structure that's reversible. So the amyloid fiber, you can think of it as an end state. It's a glue-like structure that's really not coming apart without harsh manipulation, whereas biomolecular condensate can typically form and reverse back into monomeric protein. In certain instances, these biomolecular condensates can harden into fibrils, or if there are mutations present in some of these proteins, can actually form fibrils themselves. So slight differences, but how we can target both of these is very, very similar. So at a structural level, they are different, but also really at a morphological level, they're different as well. So here I'm showing a few electron micrographs of amyloid fibrils. Now on the left are amyloid fibers derived from, again, patient-derived. So purified and extracted from a Alzheimer's patient brain. I've highlighted in red some of the pathological filaments that hopefully you can see in the cellular background here. So these are the pathological, this is the pathological form extracted from the brain. Now, this is a very, very messy prep because it's very, very difficult to purify these from brain tissue, but we can recapitulate these structures recombinantly, and that's this micrograph on the left, or sorry, the right, where here is just recombinant tau that was grown in conditions that produce pathological filament. These structures are approximately 10 to 20 nanometers in diameter, and depending on the protein, can range from a couple hundred nanometers in length to a few microns in length. Now, compare that to biomolecular condensates on the right. These proteins kind of form puncta, basically phase separate into protein and buffer. These are micron sized. They are reversible with manipulation of temperature. And here, this is TDP43 that was allowed to condensate and then detected with a fluorescent antibody. So we know the structures of these proteins. And moving to part 2, I'm going to discuss how we design and select targeting peptides to bind to these structures to arrest aggregation. Now, here is kind of our design scheme, broken into the peptide phase and the small molecule phase. Now, in the peptide phase, we work to identify adhesive segments. We do this from computational modeling, if no structure is available, to determine which segments have the propensity to aggregate, to create constructs, to solve the structure of these. If we have a structure, like in the instance of tau, we can use the structure to determine where we would like to target with our peptides. Knowing the structure, we go into our in silico design using a proprietary algorithm from the Rosetta suite. We dock a peptide backbone to different regions on the protein, and then sample different side chain identities and conformations and score these based on their predicted binding energy. Take the top binders, we evaluate those biochemically. Now we are doing that through peptide microarrays followed by aggregation inhibition assays. Previously, we would go directly into inhibition assays and go through iterative rounds of design. Once we have a targeting peptide that inhibits aggregation, and by binding to that key adhesive site we've identified, we go about screening for small molecules. And I'm going to briefly touch on this at the end of my talk. There's a few different ways we go about screening for those. Once we identify those small molecules, we then go into optimization for the development of novel therapeutics. Now, in 2011, a really important proof of concept project was undertaken in the Eisenberg lab. One of the graduate students at the time, Stuart Sievers, actually developed the first peptide inhibitors. And we've advanced from this approach, but this really is the proof of concept and kind of the foundation of what our work's based on. In this work, Dr. Sievers set about to determine or create and design peptides that inhibit tau aggregation. So here is a micrograph of fibrils of tau formed by construct of tau known as K12. The steric zipper, so in this, the adhesive segment referred to in this protein as a steric zipper, was identified as the segment VQIVYK VQVIC. Now, this protein on its own forms fibrils. The VQVIC segment on its own can form microcrystals. Now, these are needle-like crystals. The structure was determined through microcrystal X-ray diffraction. And this is the structure of it. So these are two 6-mer peptides that form what's referred to as a steric zipper. So the adhesive segments here intercalate the side chains from a very, very stable structure at the core of the protein. To design an inhibitor, a backbone was docked onto it, and D-amino acids were screened for binding. And one of those, with the identity of TLK-IVW, was found to fully inhibit aggregation. So it inhibits aggregation here, as seen by the lack of fibrils in the micrograph, but it also inhibits in aggregation assays. So in the world of amyloid, we use a small molecule reporter, thioflavin T. So as a protein aggregates into a fibril, thioflavin T binds to that fibril and fluoresces. And that's how we monitor aggregation. So here in black is the K12 construct on its own. After about 8 hours, it rapidly aggregates into fibrils. With the addition of the peptide TLK-IVW, fully arrests aggregation. Now, this is sequence specific. By taking the scrambled peptide TLK-IVW, we see no inhibition. And what's, I think, really exciting is Dr. Sievers also used EM with nanogold to actually show that these peptides are binding to the ends of the fibrils in this manner as designed. So this proof of concept is really foundational for our work and shows that by end capping a protein, a amyloid protein, you can inhibit further aggregation onto a fibril, and in this case, also prevent nucleation of fibrils. So how do we go about identifying these steric zippers or adhesive segments that we want to target? Well, there's two ways of going about that, right? On the top, the easiest way is going to the PDB and getting the structure of those if it's available, right? So we can have different structures that we can target. We can target the adhesive segments on their own, in this case, a structure of V-cubic. Or if we have other structures known, in the case of tau here, there's a recombinant fold of tau. And the most important that we were interested in, the Alzheimer's fold, can actually use this structure to identify potential areas to target with peptides. Any of these beta strands, any of these interactions, we can target computationally with our in silico design. Now, in the event that's a completely novel protein, we can run the sequence through the ZipperDB database. So again, this uses Rosetta to predict if it's energetically favorable for the adhesive segment, for a 6-peptide segment to form basically a fibril-like structure. So again, this is the structure of tau. So you can see starting at this first amino acid here of this VQ Inc segment, you see that it has a red energy here. And what this means is starting at this residue, and for the next 6 residues, it's predicted to it's energetically favorable to form a fibril-like structure. So in the case of tau here, the important segments are VQ Inc. As you can see, it's highlighted energetically favorable. And even more energetically favorable is the VQ Vic structure that was targeted by Dr. Sievers. So there's a couple ways of going about this: known structure or predicting which segments for construct creation computationally. Unfortunately for us, and unfortunately for the amyloid world, these amyloidogenic proteins are highly, highly polymorphic. The idea of one protein sequence to one structure existed for a very, very long time. that has, in well-folded proteins, that has been shown that under certain conditions, certain proteins can form different structures. In the world of amyloid, that phenomenon is magnified, especially with neurodegenerative diseases like Alzheimer's and tau. These proteins are intrinsically disordered. We don't have the structures of a monomeric form, and with current technology, that appears to be impossible to solve those structures. But we have found there have been structures solved of very, 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 very different structures of forms of tau. And here are just a few of them. In the top, patient-derived structures, so the Alzheimer's fold that I've shown in the past, as well as the PIC fold from Pick's disease. You can see that these are very, very different. We've highlighted the beta strands here, but really, although the protein's the same, the interactions between the beta strands are very, very different between these diseases. This is further complicated with recombinant tau structures, where here is just 4 of the polymorphic structures of recombinant tau. So it's very, very important for us to be able to target the correct structure. So, we use EM to verify that, but with also the very polymorphic structures, it's important to have an approach to screen these different structures for different potential inhibitors. And that's one way that we really utilize the peptide microarrays to be able to screen large numbers of peptides against different forms. So this, we see, the polymorphisms in the tertiary structure of the proteins. Again, it's further complicated with the quaternary structure of these. So these are the patient illustration of a cross-section of tau filaments from patients. In the top, you see that at the top, we can see that the Alzheimer's fold can form paired helical filaments. Through different interactions, it forms straight filaments. For PICs, it forms narrow and wide PICs filament forms. So because of this, we have to use large numbers of peptides to create and design our targeted peptides. And this is how we go about doing it. It's really a 3-step process. It starts with the design process. So here we identify on the structure regions we want to target the peptide. Using Rosetta, we dock the peptide backbone and then sample different side chain identities and orientations, and estimate the binding energy in this delta delta G function. So in this instance of this peptide KSIVFE docked to tau, we get a inhibition potential. So the predicted binding energy, we get a predicted binding energy. So we do this and create millions and millions of designs. We rank all of these based on binding energy and then go into peptide assessment. And just a quick note that although protein design and computational protein design is advancing, I would say, especially for peptides, there's kind of a poor correlation between predicted binding and effect of these. And really, what I mean by that is inhibition or true binding. So we have to screen large numbers of peptides for activity. Now we've moved on to using the peptide microarrays, where we select, the top 10 to 20,000 of these peptides to be screened, to screen for binding to tau fibrils on the microarrays, to select which peptides we want to further evaluate. And we do that through biochemical assays, so inhibition assays and specificity assays, and go through iterative rounds of optimization. And this is just one peptide that went through 3 rounds of optimization, and we were able to significantly improve the inhibition of these. But this is intensive work, both costly, time intensive, and uses lots of recombinant tau protein. So we're really only limited to a handful of peptides we can screen this way. So prior to introducing microarrays in our pipeline, we were really only limited to a couple hundred peptides that could be synthesized from our design run of millions. So we set about evaluating how can peptide microarrays really expedite this work. So for this, we took the structure of tau. We designed 3,500 novel peptides for a microarray. We screened them for binding to aggregated fibrillar tau and counter-screened against monomeric tau to eliminate any peptide that bound to the monomeric protein. From this, we found approximately 50 peptides that showed strong binding of tau amyloid to the peptide on the microarray with no bind to the monomer. From this, we took 48 peptides, we screened them for inhibition, and this is just a summary here, where the black line is the baseline aggregation or the background signal, and we found that 12 peptides inhibited aggregation in vitro of tau. So in a matter of days, essentially, we were able to design these peptides, pepperprint screened them on the microarrays. We then synthesized a handful, although we synthesized the ones that showed strongest binding and found inhibitors. And I would say this is a matter of days instead of years. And just to kind of show the power of the microarrays for our approach, I'm showing the inhibition curves of 2 peptides. On the top here is an empirical peptide that in all took a few years to develop and optimize, and we were able to see it inhibited with an EC50 of approximately 15.5 micromolar. For one of the 12 inhibitory peptides from the microarray, it's on the bottom here, this had an EC50 of approximately 14 micromolar. So by using the peptide microarrays for this initial selection of peptides, we're able to dramatically reduce the time and cost associated with peptide discovery, which is very, very, very exciting for us. So knowing that we can use this to develop novel peptides, novel targeted peptides, we performed a post hoc analysis of our peptide library. Now, the ADARx peptide library is several thousand peptides composed of natural L amino acids as well as non-natural D amino acids. So we took all of our L amino acids and put them on a microarray. And we looked at binding on the microarray, so microarray signal on the x-axis, and looked at the correlation to biochemical inhibition of the tau aggregation assay, the IC50 and micromolar here on the y-axis. So what we found from this in this post hoc analysis is there's several binders to tau, but they bind to non-adhesive segments. So we're seeing good signal in the microarray, but no inhibition at all. So they're binding somewhere, either not on the end of not on the fibril end, or they're binding on the fibril end at a location that is not essential for aggregation. So by using microarrays, we can quickly eliminate all of these non-specific peptide binders to our protein of interest. Further, on the y-axis, we see many peptides that inhibit but have no binding. Now, I've dubbed these off-mechanism inhibitors. They inhibit tau aggregation, but they don't appear to inhibit based directly on binding to tau. Right? So now we've been able to eliminate through the microarrays kind of two categories that could end up in dead ends of targeted peptide development. What's really exciting in the green box is there is a subset of peptides that are both good inhibitors and good binders to tau. So now, using the microarrays, we can funnel, instead of going directly from millions of designs into 100 to evaluate and hopefully get 1 or 2 that are inhibitors and then go through iterative design, we can now design our millions of peptides, screen tens of thousands or more in a microarray, and select the best binders for further for evaluation and aggregation assays, as well as further development. What's very, very exciting from this work is we're able to determine which adhesive segments are exposed on the tau fibrils, but not in the peptide, or sorry, not in the monomeric tau. So here's looking at kind of a summary of data from peptide microarrays to aggregated tau and monomeric tau. Each one of these colors represents a different segment on tau that was targeted. So you can see that in orange and in purple, we've identified 2 segments that are exposed in the aggregated form and form a structure that is not present in the monomeric form. So now we know, kind of narrows down on what adhesive segments we can target for specificity to the aggregated form versus monomeric form. And we can also see that there are certain regions, such as in magenta here, that is well represented in both the aggregated form and the monomeric form. So now we know that, okay, if we want that this adhesive segment is likely always present in tau, and we can't differentiate the 2 forms. So this is very, very exciting work. Now, it's one thing to be able to differentiate between aggregated and monomeric, but we asked the question, can we differentiate between different aggregate forms? And indeed, we can. We went about an experiment with the protein TDP43. And so TDP43 is linked to ALS, and it forms maybe more polymorphic than tau. So instead of just forming different fibril forms, it can form condensates as well as fibrils. So we want to see if we can differentiate in microarrays that bind to these different species. And indeed we can. On the y-axis, we see the bind to TDP43 condensates, and on the x-axis, TDP43 fibrils. Here, there are some peptides that bind both of them, but we also have selectivity to these different forms. Further peptides from this screen were evaluated, and 2 of them, peptide D450 and D399, inhibited the aggregation of TDP43. So this construct of the C-terminal domain construct of TDP43 is inhibited really quite well with these peptides discovered on the microarrays, and has greater than 15-fold specificity for TDP43 over tau. And we can actually map those differences. So here are a few of the structures that have been solved for TDP43. So here is an in vitro structure on the left, a pathological structure on the right. The condensate binders are mapped on the top, and the fibril binders are mapped on the bottom. And we can see that the primary hits between the condensate binders and the fibril binders target different beta strands on these structures. So by implementing the microarrays, we can screen large numbers of peptides now for binding. And we have confirmed that binders can be inhibitors from this. We've eliminated potential dead ends by selecting off mechanism inhibitors. And we've been able to we've been able to improve our specificity for aggregated versus monomeric form and different aggregated forms by increasing the throughput from these. So it's become a very, very powerful tool. So it's one thing to discover these peptides, but how can we develop them into a novel therapeutic? Well, say, remember back to one of my first slides, for a systemic amyloidosis, perhaps we can use a peptide directly as a therapeutic, since that is occurring in the blood of a patient. For neurodegeneration, these are all interneuronal and in the brain. And with current technology, that is a seemingly insurmountable hurdle for peptides, because peptides are very, very poor at penetrating the blood-brain barrier. What can be good at penetrating the blood-brain barrier is small molecules. So we leverage our targeting peptides for small molecule discovery, because we believe that's going to be the best way to bring a therapeutic to the clinic. And we do that in two different ways: a high throughput screen and now a DNA encoded library screen. So in the high throughput screening, we look for displacement of our targeting peptide. We conjugate our targeting peptide to a fluorophore. This peptide fluorophore is then incubated with fibrils. The peptide binds the fibril end, and then we screen for displacement against small molecules using fluorescence polarization, because you can measure the binding, and we measure the binding and release of the peptide through fluorescence polarization. So here, the competitive small molecule will displace the peptide, giving us a change in fluorescence polarization and an indication that small molecule is binding here. And we've screened approximately 750,000 small molecules in this manner and had about 0.13% hit rate. Now, this is a low hit rate, in part due to the difficulty of having a fluorescent assay with small molecules. Small molecules are great at interfering with that. So we're now using DNA encoded libraries for our small molecule screening. So this uses referred to as big data subtractive screening. So what we do is we use DNA encoded libraries. These are libraries composed of small molecules that have an identifying DNA tag to it. These libraries have 1 billion, 10 billion, or more unique compounds here. We screen against monomeric protein, fiber protein, and then fiber with a bound peptide. So here, we allow the small molecules to bind to the fibrils. They can bind anywhere on the fibril. We want it on the fibril end. So what we do to find those is we use our targeting peptides to block the fibril end. Now, anything that is specific to that, specific as an end capture to the fibril, is displaced. We sequence everything that is in this section, and then, in this tube and this tube, and the difference between those is going to be the small molecules that bound to the fibril end. And here is just a quick summary of the results from this work. From the high throughput screening, we have identified small molecules that are potent tau inhibitors. And this is thioflavin, a dose response of a thioflavin T aggregation assay. With little to no small molecule present, we see no inhibition. At high concentrations, we see no fibrils present. So we can discover small molecules through this high throughput screening using our peptides. With DNA encoded libraries, we are finding small molecules as well. So here, feature intensity is basically, you can think of it as how many times the DNA tag was read in the next generation sequencing. On this axis here are small molecules that are bound in both the tau fibril only and tau with peptide. So these bind somewhere on the tau fibril. But here on this off axis here, we see features that are more prominent in fibril alone. So we believe these are binding to the fibril end, and we're currently evaluating some of these. Further, these small molecules are active in a variety of assays. They're active in the full-length tau aggregation assays that I showed you on the previous slide. They inhibit full-length tau aggregation. They inhibit the aggregation of the adhesive segments isolated from tau, as well as they're active in cellular reporter assays. So now what we can do in our pipeline, and what we feel is really groundbreaking, is starting with a structure. We can identify the druggable site, which were previously thought to be undruggable based to kind of the flat nature of these. We can identify those structurally. We can confirm them with our peptides, and we've increased throughput using peptide microarrays. And then we can leverage those peptides into small molecule hits for future development into therapeutics. And to end my talk today, I just wanted to acknowledge everyone at Adarex, the biochemistry team, which is responsible for the majority of the work that I've shown today. They've really done some great work and really advanced both peptide design and screening work, as well as the assays and methods to evaluate these peptides, and our cell biology team who gets to either confirm we were right in the peptides and small molecules that we discovered biochemically, or tell us that we're on the wrong path. So I just want to end my talk and thank all of you for joining me today, and Pepperprint for giving me the opportunity to share just some of our work today.
Well, thank you very much, David, for a very most interesting presentation. So, so we have, I think, a little snafu. Not sure whether the Q&A function did work. We have not received any questions. Are there any questions by the audience? Would you be able to type a question if you're interested? Let's maybe give this a minute or so. So otherwise, I would suggest within the time frame, since we have already progressed past the 50-minute mark, if there are no questions, we're going to start and wrapping this up. By the way, it was very interesting to see, David, the flow, the Rosetta prediction, then the peptide screen, and the further characterization of the candidates. Interesting layout. I think here's a question. Let me see here. So one question by someone from the audience. Have you tested any of your candidates' molecules in mouse models? Can you maybe comment on that?
Yeah, I think that's a great question. So I will say that we have tested previous generations of peptides in some mouse models. There were indications that they did modulate tau aggregation. I think, unfortunately, many of the, especially Alzheimer's mouse models, are not the best at recapitulating disease. And especially in the past year or so, we've been more wary about that. And where we stand right now, at least with the small molecule work, is we still want to make sure that these small molecules, pass certain checkpoints, right? With specificity to tau or whatever protein of interest, as well as basic ADME before we really move into the mouse models. But in short, we have confirmed that the peptides do work in mouse models, and small molecules in mouse models will be performed in future work.
Great, thank you, David. So here's another question from the audience. Based on the peptide hits, can you comment on the potential safety of candidate molecules as oral agents?
Yeah. Not confidently, I can't comment on that. I would say that just theoretically, a native L amino acid would likely be a very, very poor candidate, especially orally. I've had discussions with people and some immunologists that have indicated that if you have a D amino acid peptide, it may be toxic or may have spur some kind of immune response. But I haven't personally seen any really compelling confirmation to that. And I think it's a very, very important question. And I think also point to one of the benefits of being able to screen multiple peptides in the microarrays to have backup peptides and small molecules eventually in case there is some sort of toxicity or safety indication.
Okay, thank you, David. Here's another question. So I think the time permits, maybe we'll do two more, including this one. And I read it to you. Do you find any common sequential property in the amyloid binding peptides? For instance, high aromatic content, mostly positive, negative charges, etc. Any commonalities among the different hits or molecules that you find?
Yes. So, at the computational stage, the sequences are varied and can be quite diverse. Now we do, limit what can be done. So we're what can be part of the sequence. So, we exclude methionines, we exclude cysteines, sometimes glycines. So we exclude certain amino acids. But from the Rosetta predictions, we do have a very, very diverse sequences. But I will say that through empirical design, without confirming binding, we have seen a emergence of aromatic residues, and that kind of goes to a dead end, because as you incorporate more and more aromatic residues, we see better inhibition, but also loss of specificity. So we do know these aromatic residues inhibit, but lose specificity. And when it comes to the more specific PEP residues from the microarrays, we do see certain regions in the peptides that have, maybe a preference for certain amino acids, but from the peptide screen of microarrays, they are quite diverse from each other.
Great, thank you, David. So here's our last question. Again, an attendee who already asked a previous question about the peptide hits, and he wants to know whether there are any interferences about their ability to cross the blood-brain barrier. Is anything known to that at this point?
Yeah, so there's been a lot of work done looking at how peptides can cross the blood-brain barrier. We haven't really undertaken that work ourselves. We've been more focused on, the development of these peptides as tools. So when it comes to, how can peptides cross the blood-brain barrier? Well, I would say one approach that has been evaluated by some companies is actually viral. If you have an L-amino acid peptide, induction or introducing that into the cells via a virus is one way of doing that some people are working at and may have potential in the future. When looking at just penetrance of a peptide, so a peptide cross a blood-brain barrier through, say, a infusion, there are certain sequences, cell penetrating peptide sequences that can be affixed onto the terminus of a peptide that facilitate the penetrance of a peptide into a cell. But also there's been some work and some hypotheses that some of these sequences can allow penetrance across the blood-brain barrier. But again, there really lacks, at least to my knowledge, any really magic bullet way to get these peptides across the blood-brain barrier, and that's why we've introduced the small molecule screening. So because we believe with current technology that small molecules remain the best modality for treating neurodegeneration, because they have a higher potential of crossing the blood-brain barrier.
Great. Well, thank you, David. So as we are approaching now the top of the hour, let's begin to wrap this up. So thank you all for attending this webinar presentation. And again, many thanks to you, David, for letting us in on these details. Very exciting, interesting work. So we will make the presentation slides and the webinar file available after this presentation. If you do have any questions, please contact the PepperPrint team directly. Also, feel free checking out our website for any updates, future events, and webinar announcements. We will end this webinar now. Thank you to everyone for your interest and dialing in. Thanks again, David.
Thank you very much.